Hi everyone, today we're taking notes on cell structure and function and also tools of a scientist. So please make sure you've got your name, the date, the period, and the page number filled in. It would be helpful to have a highlighter or a different colored pen or pencil so that when we highlight or underline something, it stands out a little bit. We're going to start talking about cell differences. There are a few cell differences we're going to speak about. The first one is cell diversity. The first thing that stands out in terms of diversity is the size. So depending on the cell and the organism, the cells inside will be all different sizes. So even within humans, different cells are different sizes based on their function. Cells also come in a variety of shapes. So the shape usually reflects the function of the cell. So just like with enzymes and proteins, the shape determines the function. Here in cells, the shape determines the function as well. You'll see the nerve cell has a very different shape than a red blood cell, than a muscle cell, than connective tissue. So the shape of it allows that cell to do its job. Excuse me. Another difference is the internal organization. So, cell organelles, which we already know, are cell components that perform specific functions for the cell. There are two classifications of cells we will talk about this year. Eukaryotes, okay, that E is silent, eukaryotes. These are organisms whose cells contain a membrane-bound nucleus and other organelles. So, an example of eukaryotes are humans, okay, anything, plants that have a nucleus that's covered with a membrane and other membrane-bound organelles. Membrane-bound organelles include like the Golgi body, the endoplasmic reticulum, whereas prokaryotes are unicellular, so uni meaning one, so unicellular organisms that lack a membrane-bound nucleus and lack other organelles. So prokaryotes do have genetic material, but it is not contained within a nucleus, so the DNA just floats around. And sometimes you will find ribosomes in prokaryotes, but ribosomes are not membrane-bound. Next, we talk about levels of organization. For us, where you need to start in terms of knowing the levels of organization is here at the cell level. So cells make up tissues, a bunch of tissues make up organs, organs make up excuse me, organ systems, and organ systems make up organisms. You will have to know, sometimes questions will ask it to you from smallest to largest. Sometimes questions will ask from largest to smallest. When you're all set on this side, please turn to the back of the front page. This is where we start our Tools of a Scientist section. So, as we start talking about cells, we will also start using microscopes. Okay. So here, the microscope is the most common tool used in biology to see those things that we can't see with the naked eye. So the microscope is an instrument that produces an enlarged image of an object. Magnification. We use magnification to increase an object's apparent size. So this means we're making it look bigger, but we're not actually making it bigger. So that cell may appear bigger, but it doesn't actually change size. And the resolution is the power to show details clearly. Okay, next up, when we talk about the compound light microscope, that's the one we will be using in class. We do need to be able to find the total magnification. So to find the total magnification of the microscope, you must multiply the magnification of the eyepiece with the magnification of the objective you are looking through. At this point, if you would highlight, circle, star, underline, total magnification, so you know where to go back to if you need to have a little refresher on this. So we have an example. The eyepiece of a microscope is 10x, so this x is telling you there's a magnification. You need to have the x after the number. The low power objective is 4x. Okay. And the magnification of the microscope when viewing under low power is, so you would do 10x times 4x equals 40x. So you want to make sure you still have that x after your number. So if you were using a different objective, you'd multiply the eyepiece by the different objective. Next up, we talk about what your image will look like under the microscope. So when an object is placed under a microscope, its image is flipped upside down and backwards. So you'll see if you put this letter E, lowercase letter e, on the stage of the microscope like this, you will look at it and it will look like this. So it will flip upside down and backwards. When you're all set on this page, please flip to the next. We will start talking about the field of view. So the field of view is the total area that can be seen under the microscope. The field of view, so on the microscope we usually see a circle when we look into it. So this is your field of view, abbreviated with FOV. This field of view decreases in size when you go from low to high power. So the more you magnify, the less you see. But what does happen is your image gets larger. Okay. Next, we review how to bring a specimen into focus under high power. So there are 6 steps. You always want to start on low power. Okay, so if you have a highlighter or a different colored pen, highlighting this low power piece would be a nice idea. We always want to start on low power. You adjust the coarse adjustment, which is usually the bigger knob, to get it into a good focus. And then you can use the fine adjustment, or usually the smaller knob, to get it really crisp and clear. Once you have your specimen arranged, hopefully in the middle of your field of view and on low power, you can sometimes go right to high power. Taking a second to go to medium power and focus it there would not be a bad idea. When you are on high power, you only want to use the fine adjustment. The high power objective is probably almost touching, if not touching, the cover slip of your slide. So if you're using the coarse adjustment, it can go too far down and crack the slide that you are using. Okay. Lastly, when you're done, you can adjust the diaphragm if you need a little bit more light. If it's a little too dark, adjust it, open up the diaphragm to get a little more light through so you can see the image a little brighter. When you're done, please flip to the next page and we will review the parts of the microscope. So this is your microscope. You will see different microscopes, different pictures of microscopes, but this is one example with all of these labeled. You'll see the parts labeled here, and on the left-hand side, they are defined with their function at the bottom. You will need to know how to label the microscope, you will need to know the parts, and you will need to know what they do. That's the end of our notes today. Thank you so much.